We are going to be talking about cell cycle regulation today. So we talked about what the cell cycle was, and then we went through the process of mitosis. Yesterday, we talked about the evolution of mitosis and the process of binary fission. So if you could get your notebooks out, I believe, if my calculations are correct, I believe this is the last day of notes this semester in AP Biology, which, woo, very exciting. Tomorrow will be kind of more of a biography storytelling activity day. But today is about cell cycle regulation. This is the last bit of content for the first half of AP Biology. If you remember from the beginning of the year, the stuff that's in the beginning of your notebooks, let me grab it really quick. There are 8 units that are covered on the AP exam. We have done the first 4 of them, which means we are on track for the end of the year. We've talked about the chemistry of life, cell structure and function, cellular energetics, and now cell communication and the cell cycle. Um, we will start next semester with heredity, go into gene expression and regulation, and then hit natural selection and ecology at the very end of the year. So we're going to kind of stick to the small stuff a little bit longer and then kind of zoom out to big picture stuff towards the end of the year. But we have finished almost half of AP Biology, which is insane. If you think about it, that means we are a quarter of the way through a college semester, which is also fun. Because this class is a college course spread over the course of a year. So it's a lot of fun. So today, my goal for you is to be able to name the roles and functions of different signaling molecules throughout the cell cycle. Um, to start us off, what the cell cycle, there's kind of a well-known allegory, that's the word I'm looking for how the cell cycle is controlled. So the life cycle of a cell is operated or controlled by this set of molecules that operate in a cycle or cyclically that will trigger and coordinate key events for the cell. And one of the most common reference points here is a washing machine. If you've ever interacted with not one of the new ones with the fancy buttons that sing a little song whenever they're done, but the old school washing machines, if you start a load of laundry and you twist that knob to wherever you need to go, whether that be regular fabrics or heavy fabrics, and you have different levels of what you would wash them for, and they say, okay, now we are in the agitating cycle, now we are in the spin cycle, now we are done. You've got that kind of arrow that moves with the washer. The way it works is that it won't move to that next step until it receives a signal, right? So whenever you start a washing machine and it starts to fill up with water, at least the older ones would fill up with water, it won't move to the next step until the weight, the barrel of the machine hits a certain weight, or that water hits a certain level. And when that water hits a certain level, it will move on to the next part of the washing cycle. That's the way it works with the cell cycle. We have to wait until the cell has all of the appropriate things to move to the next one. So there are 3 big cell cycle checkpoints that we're going to talk about that act as those little okay, we're done with this step. We have everything we need. Time to move to this step. I kind of lost myself in the metaphor there. We okay? All right. So I mentioned these cell cycle checkpoints. A checkpoint is a control point. This is where we get our stop and go signals that will regulate the entire cell cycle. If you remember the cell cycle from Monday, it is Thursday. we drew this little diagram right here. We said there were 4 phases of the cell cycle: G1, S, G2, and M. Um, and there are certain checkpoints within this cell cycle that we have to hit in order to have a successfully operating cell cycle. The first one is in G1. So it is right at the end of G1. That is our major decision checkpoint. I call that the to divide or not to divide checkpoint. That's the one where our cells say, okay, do I need to replicate? Can I replicate? Sometimes that doesn't happen. Sometimes they don't get the proper signals. And that's when they will go into this kind of pocket universe of G0. And we'll talk about G0 later. But if the cell gets the appropriate things needed to say, okay, yes, I need to divide, it can then move into the S phase. If you remember, the S phase stands for synthesis. So it'll duplicate its DNA, and then we'll go through G2. And this is my favorite checkpoint. This is the G2 checkpoint. I like to refer to it as the phone keys wallet checkpoint. You know, when you're headed out the door and you're like, okay, do I have everything? And you kind of slap your pockets, make sure you have your phone, your keys, and your wallet on you. That is what the cell is doing in the G2 checkpoint. Do I have undamaged DNA? Do I have 2 copies of it? Do I have enough organelles to make it through this process? Do I have the right signals? Do I have everything I need for mitosis? And if it does, if it finds all its stuff, it can head into M phase and out of interphase. And we also have the M checkpoint, or the spindle checkpoint. I don't like the spindle checkpoint because it makes it seem like there's one in S phase, because S checkpoint. I like the M checkpoint better. It is in metaphase, and you know, in metaphase, all of our chromosomes are lined up down the middle, and all of our little spindle fibers have connected to those kinetochores on our chromosomes. It is then we have another checkpoint to make sure all of those kinetochores are connected to those spindles. Because if one of them takes off before everything's connected, cells wind up with different numbers of chromosomes. It's like the annoying little beep your car does when you take off without your seatbelt on. If you're not connected, it's going to send a signal not to go. Make sense? All right, those are our cell cycle checkpoints. Another way we like to reference the cell cycle is as a clock, right? I like this metaphor because it is very much, it helps us visualize this process. This isn't just like I get this step done, I get this step done, I get this step done. It is a gradual movement of cell components and cell signaling molecules, like a clock. The big hands and the small hands on the clock don't just go, okay, it's 6 o'clock, it's 7 o'clock. We see the movement, the slow, gradual movement of time, and we see the slow, gradual movement of the cell cycle. And there are 2 very important molecules. Oops, there's a door right there. Watch out. There are 2 very important molecules that are in charge of this kind of clock, making sure that a cell gets the proper signals. They are protein kinases. If you remember from last week, protein kinases are enzymes that phosphorylate proteins. Yes. I forgot the word proteins in there because I got too ahead of myself. Protein kinases are enzymes that activate and deactivate proteins by phosphorylating them. Woo. So we talked about these in phosphorylation cascades, right? When we had one protein kinase that phosphorylated another protein kinase that phosphorylated another, and so on and so forth. These also come back in the cell cycle because the cell cycle depends on cell signaling. And then we have cyclins. Cyclins are a special protein that will activate certain kinases, specifically cyclin-dependent kinases, or CDKs. So we've got our protein kinases, or our cyclin-dependent protein kinases, and we have our cyclins, which are our little activation molecules. One small little sort of looking for detail that I want to kind of make you aware of here is that the activity of cyclin-dependent kinases increases and decreases with the concentration change of cyclins. That sentence kind of has you infer that the concentration of the kinases does not change. It is the activity of the kinases that change. So there are always cyclin-dependent kinases chilling in the cell. They're always there. We got a lot of stuff floating around in the cytosol. So the activity of these kinases will change as the concentration of the cyclins change. So when we are receiving these signals, we aren't changing the concentration of the kinases. We're changing the concentration of the activator for the kinases. Questions here? All right. It's kind of like working at a fast food place, right? You're always there. There's food always there, but you're not doing anything unless there's a customer, right? An activator, unless you have a time to lean, time to clean manager, which we try to avoid. All right, so these are our cell clocks, and I've got some graphs to help you visualize this. The first one I want to point out, and I'll grab my little highlighter. The first one I want to point out is this one over here, because we have our cyclin-dependent kinases, and the cyclins, of course. Let's see, there we go. Yes, please. There we go. So this graph right here, of course. Why do I do this to myself? There we go. This graph right here. Up here, this dotted line, that is our concentration of our cyclin-dependent kinases. They are always present in our cell. It is the concentration of the cyclins, this line down here, that changes throughout the cell cycle. And we can see that cyclins will gradually increase in concentration in the cell, reaching an apex, or that kind of top point, in mitosis, or in M phase. So as the cell is preparing for division, we're ramping up and we're ramping up and we're ramping up the concentration of cyclins. And as the cell divides, that concentration plummets. Because we've done division very recently, we are currently doing division. We do not want more signals to trigger another division phase if we're not ready for it. So we break down that concentration. So we're in G1 again, and then we increase and increase and increase our cyclin concentration until we get to M phase, and boom, as we divide, it plummets again. All the while, our CDKs, our cyclin-dependent kinases, they, in concentration, are going to stay the same. But if we wanted to measure the CDK activity, if we wanted to measure the cyclin-dependent kinase activity, odds are it would follow the same pattern because it is the cyclins that activate it. That makes sense? All right. This one over here introduces us to another factor. It is the maturation promoting factor, another little signal that pops up in mitosis. So our cyclins, their concentrations, again, are going to increase until it hits M phase, and they're going to plummet. And they're going to slowly increase until we get to M phase, and we're going to plummet. We have another factor, which is an M phase promoting factor, a factor that helps the cell know, okay, yes, it is officially time to divide. And we see it spike up in M phase, and as soon as we're like in the throes of M phase, it plummets again. And it is gone from the cell until we start M phase again. Questions here? All right. So I want to kind of talk about some of these checkpoints and specifically the signals that they respond to. The signals that they respond to. So if a cell doesn't receive the go-ahead signals in the G1 checkpoint, remember the G1 checkpoint is that big decision checkpoint, the to divide or not to divide checkpoint. If they don't receive the, yeah, go ahead, we need to divide, they will enter a non-dividing phase. So there's kind of, I like to think of it as a little pocket universe, but it's just kind of a step outside of the cycle called G0. G0 is a resting state. The cell is still doing its normal cell activities. It still has a metabolism. It still sends and receives signals. It still does everything a normal cell does. It's just not dividing. But just because a cell enters G0 does not mean that it's going to live its entire life in G0. Because a cell, one of these G0 cells, can be triggered and called out of G0 by external cues. Specifically, growth factors is one big example. A growth factor is just a signal that is sent by a neighboring cell. Their goal is to stimulate cells to divide. Growth factors are super duper important in the processes of healing and the processes of development. So if you ever take a developmental biology course, you'll talk a lot about growth factors because their concentrations kind of decide if a developing organism will like reach maturity. But another one is with healing. So if you are like me and you are a very clumsy person and just kind of live your life without regard to furniture, you get cuts and scrapes and bruises on a regular basis. When that happens, the cells around those cuts and scrapes and bruises send out these growth factors. They say, hey, something got damaged, we need to go fix it. So if you get a paper cut, right? All of the cells around that paper cut are like, hey, we lost some cells are damaged, we need to fix that. They will release growth factors, and then cells will be triggered to divide more and hopefully patch up and replace those damaged cells. So this is G1 and those little G0 signals. Questions here? So that's our signals for our G1 checkpoint. We're also we're not going to talk about each checkpoint specifically, but there's a couple signals that I need you all to know about. The other one is the M phase checkpoint. So this right here is an image we know and love. We have drawn this image. It is a cell in metaphase. After metaphase comes anaphase. Anaphase will not happen until all of our chromosomes are all attached to the mitotic spindle. When our kinetochores are all attached to the spindles, it will activate our regulatory molecules. Those regulatory molecules are like, okay, we've got the right signal. Everybody's strapped in and ready to go. Let's start breaking down these microtubules. Let's start building up these lengthening microtubules, and let's get anaphase underway. And we know that anaphase is the fastest stage of mitosis. So once that signal gets sent, the cell is in go, 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 go mode. So this is our M phase checkpoint signaling molecules. They are heavily involved with those kinetochores and whether or not they are attached properly to our spindle, whether or not our chromosomes are buckled in and ready to go. This signal is very important because if we did not respond to this signal, some of these chromosomes might not get properly separated. Some of them might get lost or damaged, and we desperately need all of our chromosomes, all of our genetic information to make it through this process safely. So that's our M phase checkpoint. There are also some external signals that I kind of want to talk about. The first is density-dependent inhibition. Cells know when they get crowded, right? And when a cell gets super crowded, gets to that overload point, they will stop dividing. It's like if you're in a very, very crowded room, odds are you will not invite more people into said crowded room. Right? So these cells, if they are too crowded, they will stop dividing. They're like, okay, we're good. We don't need any more of these. Let's stop. There are also some anchorage dependent signals, and I'm not talking about Anchorage, Alaska. I'm talking about being anchored to a wall, to a matrix, to something. A lot of cells won't grow unless they feel stable. Same. Right? We don't want, if we aren't attached to something, if we don't have that stability, it makes it really hard to feel safe enough to grow. If a cell can't ensure its progeny's future because it doesn't have something to be attached to, it won't divide. So some cells have to have a secure anchoring point, whether that be some sort of extracellular matrix of tissue or a wall or something like that. That is why a lot of bacteria like to hang out on your teeth, because they are a strong wall of security. So these are some external factors that can affect the cell cycle. Now, all of this stuff that we've been learning about the last couple weeks has been super intricate. And if you think about how all of those intricacies over millions and millions of years created and led to and eventually changed into what we are learning about now, it can get like, oh wow, we are really good at being alive. We are really good at having our cells function properly. This process is not perfect. In fact, a lot of cells processes are super inefficient. And I think we'll talk about that a little bit more next year. Next year, I guess it is technically next year. But this cell cycle is all dependent on a bunch of proteins. And proteins are dependent on genetic information, which is all just on a molecule of DNA. If you remember from freshman bio, or if you took bio 2, that means all of this stuff, all of these proteins are coded by a bunch of A's, T's, G's, and C's on DNA. And we'll talk about that with heredity next semester. So if one of those letters changes, or if something happens to one of these cell cycle regulator proteins, stuff can break bad quick. Usually, our cells can detect, hey, something's wrong. Let's hit the self-destruct button, and they go into apoptosis. Sometimes, if the cell signals for apoptosis, right? We talked about our different types of apoptosis processes and how they have all of these different signals. If a gene for one of those apoptosis signals is damaged and we don't get those apoptosis signals, that can lead to loss of control of the cell cycle, which can lead to cancers. So all cancers are just a loss of control of the cell cycle. So the first line is just a hypothesis. I've got some important information down below it. This hypothesis, whenever we are looking at what a cancer cell is and what it does, cancer cells might need, or don't need, that's what it is. It's supposed to be don't. Cancer cells don't need growth factors. They make their own, or they have an abnormality in a signaling pathway that constantly triggers the growth factor signal. That's the hypothesis. A lot of the time, and we've gotten very, very good over time at studying what cancer cells do and how cancer cells happen. We still don't know everything. So this is one hypothesis about why cancer exists. We do know that if cancer cells do stop dividing, they do it randomly. It's not like they get to that G1 checkpoint and they're like, oh, I don't have what I need, I'm going to stop. They don't listen to those checkpoints. If they stop growing, it's just wherever they decide to stop growing. If we take cancer cells and culture them, so grow them in a lab, they, odds are, will divide indefinitely. And tomorrow we get to talk about the cells that allowed us to do all of this research in the first place. The first human cells that were able to be cultured in a lab were cancer cells. When these cells divide indefinitely, we say they have undergone transformation. And that is our kind of notification that those cells have become like cancer cells. So there is some sort of transformative process that cells undergo where they stop listening to cell cycle regulation and just do their own growing, constantly and with reckless abandon. So, in the body, any cell has the potential to become many cancer cells. Most cancers start off as one cell that didn't listen to apoptosis signals. Typically, these cells have altered proteins on their surfaces because if we're changing up proteins and something's going wrong, odds are we're going to hit one of those membrane proteins. And that can activate an immune response. If something changes on the outside of a cell, our immune system is very good at saying, hey, that's not us. That's not supposed to be here. Let's destroy it. If that immune response doesn't happen or doesn't succeed, it can proliferate and form a tumor. Not all tumors are cancerous. Benign tumors, they're just kind of some cells got lost in translation, got the wrong signals, messed up a little bit. They stay where they are. They don't spread. They is just like a mass of cells that grew where they weren't supposed to grow. If they're in places that are easily accessible, usually we can just go in, grab them, and be done with it. Sometimes, if they're not doing anything negative, we just leave them in there. However, a malignant tumor or a cancerous tumor will spread to new tissues and can even move to different parts of the body and typically impede cellular functions. These cells are our transformed cells. So I've got these two little images up here. A malignant tumor, we've got this big mass of cells. We also have these cells kind of moving out and spreading. Whereas a benign tumor, they just kind of grow in their little pocket and do their own little thing. Unless they are compressing an important part of the body or impeding like a tissue function, they typically aren't that difficult to remove, but sometimes they're just in inopportune places. So 2 different types of uncontrolled cell growth. However, if they can spread, if they can move to new tissues, we call those cells cancerous cells. Questions here? All right. So, these cells, these cancer cells, just like any other cell, they send signals. And cancerous cells, particularly invasive cancerous cells, they are really good at trying to get the resources they need. So, they can actually secrete signals that tell blood vessels and lymph vessels or lymph node vessels to grow toward the tumor, therefore supplying it with the resources it needs and giving it a super highway to the body. If cells break off and enter those vessels, they can move to other parts of the body and set up shop. And that process is called metastasis. It is where cancer cells move to a site that is different from their original growth site and set up a second or third or fourth or fifth kind of zone. Yes. I wouldn't put it past me. Oh, no, I think I just used the wrong form of secrete. Don't apologize. This is obviously my bad. Yep, that's it. One extra letter. So, these cells can send off signals through the rest of they can send off signals in their kind of neighboring area, and in those signals, sorry, I clicked on my computer, so I have to go back. trigger our blood vessels and lymph vessels to grow toward it. Therefore, vascularizing it, giving it like all of the resources around it, and giving it access to the circulatory system. Which is a lot of the time when someone goes to a breast cancer screening, they check the lymph nodes under the armpits because those are the closest lymph nodes. And in order to stop the process of metastasis, we have to ensure that our lymph vessels aren't growing toward whatever cancerous mass has formed. So when people go in for cancer screenings, lymph nodes are like a lot of the times one of the first things that's checked. Also, they play a really big role in immune responses, which is why lymph nodes get swollen when we get infections. So lymph nodes swell all the time, but we gotta check them. So, cancer cells can send signals to kind of change body layout to their advantage, which, when we think evolutionarily, is pretty amazing, but also just like amazing in the worst possible way. Now, we know all of this stuff about how cancer cells interact with one another and interact with the body, which means we've gotten pretty good at treating it. We don't have, there are some cancers that are harder to treat than others. But these are our like big three treatments. The first one, I don't even have on there, but it is just like straight up removal. Go in, get as much out. And if you can get all of those growing cells out, you're good, right? Because it is just that one cell that went haywire that caused this process. So if it is still early and we haven't moved yet and we haven't spread yet and we haven't gotten through the vascular system yet, we can just go in, pluck it out, and if we don't leave any cells behind, then we got it. High energy radiation. So radiation is something that damages DNA, which is why radiation is a carcinogen, right? It's why we wear sunscreen when we go out in the sun. The sun gives off radiation. If we wear sunscreen, we protect our DNA from radiation. Therefore, hopefully, trying to reduce the possibility of skin cancers, because skin is the cells that like are exposed to the sun's rays. If that DNA is damaged, then we can eventually get cancers. But if we can take radiation and damage the cancer cell's DNA even further to the point where they can no longer function, they're gone. So we use radiation. Chemotherapy is like kind of the most well-known treatment for cancers, and the way it works is super interesting. Chemotherapy interferes with certain steps of the cell cycle, specifically in cells that divide very quickly. So if cells divide really, 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 really quick, chemotherapy goes in, they're like, hey, stop that process, interferes with those cell cycles, and those cells stop dividing and can even die off. That's why people who undergo chemotherapy lose their hair and have a lot of stomach issues, because hair follicle cells grow really, really fast. And chemotherapy doesn't know the difference between a fast growing hair cell and a fast growing cancer cell. The cells on our stomach linings grow really fast, or because they're constantly exposed to a lot, like a really high acid content, right? So we got to replace those on a regular basis. If those cells are replicating really fast, right? Chemotherapy doesn't know the difference between a cell in your stomach lining that is dividing really, really fast or a cancer cell. So it's kind of just like a buckshot, like get it all. Anything that's going fast, we cut it out, hopefully slowing the growth of cancer cells. Some cells are some treatments are actually more targeted, and we are like, this is like groundbreaking med tech right here. We are slowly but surely entering the age of personalized medicine, and this stuff, specifically targeted cancer treatments, are kind of where that all started. So breast cancer cells, some of them have a special protein on the outside of them, and if we can train our immune systems or figure out a way to introduce something to the body that will destroy cells with that external protein, with that little ID tag, we can hopefully just target those cells and none of the cells around it. A lot of the time, it is not just one of these. A lot of the time, treatments are specialized per person, and will kind of pick and choose their pieces. When I was little, my mom had thyroid cancer. She's fine now. But what they did was they went in, they removed everything, and then treated with radiation just to make sure they got everything behind. And those two combinations of things worked. She's been in remission ever since. I was like 8. So, this kind of treatment process differs from person to person, differs from cancer to cancer, and can kind of be a pick and choose board. Questions here? All right. This was a very, very heavy way to end notes in AP Biology first semester, but that's where we're going to end it. Kind of hopeful with the idea of like personalized medicine. It's super big right now and like really, really cool. And when we talk about genetics, we might get even a little bit more personalized there. Questions about anything cell cycle, cell signaling? Cool. All right. So we still have like 10 minutes left. I don't have an exit ticket for you, but if you didn't finish the exit ticket from yesterday, now might be a good time to do it. Tomorrow is my last day to receive like assignments from the semester. So if you've got like one random one floating out there, feel free to knock that out and send it my way. Tomorrow we get to learn about a very, very important woman who is kind of the reason science is where it is today. Just fun.